Have you ever thought about your future? Do you see yourself in a house of your own, with the job of your dreams and a partner who loves you? Maybe children that you can throw a ball with, or in our case, share your love of biology with.
But have you ever thought about the fact that you might not get the chance? It might seem rare, but infertility affects 15% of couples. Once you get this news, you might think that you have no options, but thanks to enormous improvements in medicine and fertility, scientists have found a way of increasing the chance of pregnancy in infertile couples through a process called in vitro fertilization.
In vitro fertilization is a process that improves the chances of fertilization by inducing ovulation in a woman in order to collect an egg, and then combining the sperm from a man and the collected egg in a culture dish. Normally, this can produce anywhere from 5 to 12 embryos. Doctors then decide which embryo is most viable for implantation. This embryo is then implanted into the woman's uterus.
In America, 18 to 45% of IVF cycles using the woman's own eggs result in pregnancy, depending on age and other factors.
Through analyzing clinical data from 665 in vitro fertilization cycles done in 2005, a study by the Stanford University Medical Center determined that there are 4 main factors important in determining if a woman would become pregnant. Out of the 30 variables analyzed, the factors with the most correlation to successful in vitro fertilization cycles were total number of embryos produced, number of 8-cell embryos, percentage of embryos that stopped dividing and would die, and the woman's follicle-stimulating hormone level. Using these factors, the study could predict whether or not the in vitro fertilization would be successful with 70% accuracy. This finding is incredibly important for in vitro fertilization counseling because it shifts the clinical focus from finding the perfect embryo to identifying methods to help the mother change her lifestyle to make the in vitro fertilization cycle more likely to work. It gives a more customized probability to each couple on whether or not in vitro fertilization will work for them, and therefore whether or not it is worth it for them to spend the money and go through the emotional toll that in vitro fertilization can be.
Keeping these factors in mind, let's talk about the current IVF therapies. For women who are generally infertile because of endometriosis, a disorder that causes the uterine lining to grow on the outside of the uterus, the strategy employed is to treat them with a GnRH agonist to increase GnRH in the body for at least 3 months before starting the regular in vitro fertilization. According to a study by Surrey and others, the aforementioned process results in significantly higher pregnancy rates than other ovarian hyperstimulation techniques. Other therapies include freezing the embryo and then thawing it when the woman reaches the perfect time in her cycle, and culturing embryos for 5 to 6 days to a blastocyst, meaning that the embryos become 8 cells before transfer to a woman. According to a study by the University of New South Wales, frozen embryos are just as successful as fresh embryos in percentage of ongoing pregnancies produced. All of these therapies are for infertile women, showing the lack of development of therapies for infertile men. Until now.
According to a study done by Zal and others, 1/3 of infertility cases are traced to men. The main reason that males are infertile is that their germ cells are unable to divide properly by meiosis into functioning sperm cells. This study used mice embryonic stem cells to create sperm-like cells that were fully functioning and were able to produce fertile offspring through in vitro fertilization. The embryonic stem cells in mice were exposed to chemicals which persuaded the stem cells to change into primordial germ cells. The natural tissue environment of the germ cells were replicated by exposure to sex hormones, testosterone, and testicular cells. The embryonic turned germ cells can undergo meiosis, which will result in sperm-like cells that have the correct DNA and chromosomal content. These cells were then injected into a mouse egg, then implanted into the mouse's womb, just like in human in vitro fertilization. This advance shows that the need for functional germ cells in males in the future is not as important as it once was. If this finding can be successfully repeated on primates and then eventually humans, the rate of successful in vitro fertilization should improve drastically. But what are the risks of in vitro fertilization right now?
It costs a lot of money, and there is no guarantee that it will be successful. A recent article released by Forbes says that the cost of one fresh in vitro fertilization cycle before medication is $12,000. Medication is typically another $3,000 to $5,000. According to Hansen and al., the risk of birth defects doubles with in vitro fertilization. There is also a risk of multiple births, which is dangerous for both mother and child and can lead to death.
Although there are valid risks associated with in vitro fertilization, the process is many couples' only hope for reproductive success. It's a sad truth that no matter who we are, at some point in our lives, most of us will either know someone affected by infertility or be affected ourselves. Thankfully, in vitro fertilization and its combination with stem cell research give those who need it a second chance at becoming a mother or father and being able to share their love of biology with their own children for many years to come. Because, yay biology!